Hi, I'd like to explain how we use a ATP monitoring machine. It's very simple. This machine is set up to read light, and it reports in relative light units. We can swab a surface with a very specific swab and then place it into the machine and get a result within 15 seconds, and it reads it as relative light units. What that actually means is it detects an enzyme called ATP, which we find in human and bacterial cells and cells of other microorganisms. If we swab a surface and then read it in this machine, it will equate the level of ATP to relative light units. So it sort of signifies how much contamination is present. We use this machine to screen the surfaces in a whole range of industries, but specifically the food industry, and also infection control when we're trying to swab if the surface is clean. Very, very simple to do, a very rapid technique, not always better than actually looking for microorganisms themselves, but a very good alternative. Okay, so I've got a whole range of just everyday items here. I've got my key fob, my specs, I've got a computer mouse, a couple of pens. And all I want to do is I just want to see whether or not these are clean or contaminated. I am assuming they're going to be contaminated because I cannot tell you the last time I cleaned these. So I would expect there to be a relatively high number of microorganisms present on these items. And alongside that, once I put them in this machine, I would imagine I'm going to have quite a high reading or a relative light unit. So let's have a look at pens. Everyday objects. What do we do with pens? Shove them in our mouths a lot. Lots and lots of people. We pass them from one person to another. Everyone borrows each other's pen. So do we clean pens? Can't remember the last time I cleaned a pen. So let's just have a look. Let's just see how many organisms are present on this pen. Bearing in mind that we're using the ATP monitor, so we're not actually counting organisms, but we're counting relative light units. And this is equivalent to ATP, which is an intracellular enzyme present in all living cells. If I was to send this swab to the laboratory to have the organisms cultured and grown, it would take at least 24 or 48 hours. But using this technique where we're looking at relative light units, which is equivalent to ATP enzyme, which we find inside cells, we can equate the numbers in general terms to microorganisms. On this occasion, there are 5,682 organisms present on this pen. That's very heavily contaminated. And just imagine if you're putting that in your mouth. That's nearly 1 × 10 to the 4, it's nearly 10. Oh, lots and lots of bugs. 5,682. So what I will do is I'm going to clean this now. I'm going to spray Steril-eeze on it. I want to give it a good clean to reduce any grime. And then I'm going to re-spray it with Steril-eeze. I'm going to leave this at least 30 seconds or 5 minutes, so I'll be back soon. So let's see what effect the Steril-eeze has had on the pen. If you remember, we had over 5,000 organisms on this pen, the one I frequently touch, pass on to friends and family, and usually sometimes ends up in my mouth. Look at that, none. That has been so effective on that pen. That's amazing. That is 99.99% reduction. This is the level of reduction that is claimed in the laboratory when they do the surface test. So again, it's proved that it's effective here. So finally, I think we'll just look at one other thing, and I think we'll look at my specs. So I'm dreadful for my specs on my hair. I often do this. As a microbiologist, you wouldn't think I would put things in my mouth, but here you go. So hopefully I'll have a nice contaminated set of specs. So let's just see how contaminated it is. So I'll swab the edge. I've had this in my mouth, if you remember. So swab the edge. I wonder how many we'll have here, because normally within your mouth you have lots and lots of microorganisms, and as you saw, I was sucking my glasses and chewing them, so we'll just see how many bugs we've contaminated my glasses with. So there we've got 2,926, so high numbers of organisms. So what I'm going to do now is just look using Steril-eeze to see how effective it is at cleaning my glasses. We'll take that swab out. Spray my glasses. I don't know how many of you do this. These are just cheap reading glasses. I'm not very good at cleaning them. I know sometimes friends borrow my glasses when they can't read. Not that they put them in the mouths, but I am dreadful for putting glasses in their mouth. So I've given them a really good spraying with Steril-eeze. I'm going to leave them for about 5 minutes just to let them dry and just see what actually happens. So I've been leaving my glasses for about 5 minutes. I've actually sprayed them and cleaned them with Steril-eeze, which is a new product containing hypochlorous acid. It's a very friendly product. It doesn't actually hurt things like spectacles or anything like that, or any plastic. So it's a nice, easy cleaner to use on the surface. So I'm just now going to swab the edges of my glasses, which is what I had in my mouth in a previous video. The reading on the previous video was 2,000 relative light units, which is equivalent to approximately 2,000 cells. So let's just see how effective this product's been. It's passed a lot of standard laboratory tests, and often when you use them in practice, you don't always expect to see exactly the same sort of result. But this is amazing. That's reduced the organism numbers on my glasses, which were 2,900. So that's a 99.99% reduction, because the result here is 0. So there is no ATP at all left on my glasses. So cleaning them, leaving them for 5 minutes in the presence of Steril-eeze, it's given a 99.99% reduction.